In this video, we're going to finish our exploration of meiosis by looking at the second half of the process of producing gametes, which, not surprisingly, is called meiosis 2. Now, it should be noted that we have just finished meiosis 1 here, which means that we actually already have 2 daughter cells that exist as a result of the first half of meiosis, meaning that everything that we're about to see actually happens twice. So after telophase 1, this is where meiosis 2 occurs, and as we will see, the process of meiosis 2 is actually quite similar to the process of mitosis. In fact, it's mainly the starting conditions that differentiate the two from each other. Namely, between the end of telophase 1 and the beginning of meiosis 2, there is no interphase that takes place in between. So even though interphase, we remember, is the stage in between cell divisions that occurs in mitosis, this doesn't need to happen again because we have already duplicated our DNA. In fact, this is the problem that necessitates a second half of meiosis. The other obvious difference between meiosis 2 and mitosis is that the 2 daughter cells that we produced at the end of meiosis 1 are actually haploid, meaning that each daughter cell only contains one homologue of the maternal and paternal chromosomes, and which one each daughter cell contains depends on how the chromosomes independently assorted back in meiosis 1. So why is meiosis 2 even necessary if we are already haploid? The problem is that even though we only have one homologue of each chromosome in each of the 2 daughter cells, there are actually 2 copies, as we can see from the fact that the chromosomes are X-shaped, indicating that the DNA has still been duplicated. So the ultimate goal of meiosis 2 is actually to take our copy number and decrease it from 2 copies of each chromosome to 1 copy of each chromosome per daughter cell. Other than that, pretty much every stage of meiosis 2 occurs identically to the way that it would in mitosis, namely that the DNA already being condensed never decondenses between meiosis 1 and meiosis 2, so the only 2 things that are necessary to split up the DNA is, number 1, for us to reactivate the centrosomes to get them to start producing the microtubule spindle fibers that are necessary in order to split the chromosomes in half one more time. And of course, if the nuclear membrane remains in place, then this would not be possible, so the nuclear membrane needs to disintegrate once more in order to transfer the copies of each chromosome to opposite sides of the cell. Once all 3 of these stages have been accomplished, it's time to advance to metaphase 2. And this is where meiosis 2 looks superficially similar to mitosis, but with one key difference. So here we can see that the chromosomes are aligning on the metaphase plate exactly as they would as if they were in mitosis in a single file line. Now, one obvious difference, as we can see, is that we have hybrid chromosomes. So remember, as a result of genetic recombination or homologous recombination, also called crossing over, our paternal and maternal chromosomes are not 100% paternal and maternal. They contain a mixture. Now, what this means is that there are actually multiple different ways that the chromosomes can align on the metaphase plate. And this diagram right here actually shows us both possibilities. So as with metaphase 1, during the process of independent assortment, there are actually 2 possibilities for what we call chromosomal alignment. And again, this word is specifically chosen here. For example, if we consider this to be chromosome 1 here, this is one daughter cell and this is the other daughter cell, we can see that the percentage of the DNA that is paternal is mostly on the top half, and then we have the second copy of the chromosome that contains a mixture on the bottom half like this. Now, this chromosome, this homologue, does not actually need to align like this. Instead, the mostly paternal half can align on the bottom, with the hybrid half aligning on the top like this, where the two copies of the chromosome would separate differently depending on which half is on the top or the bottom. So what this results in are different possibilities as to how daughter cells would form depending on which copy of the chromosome migrates to which side of the cell. And this also applies to the other daughter cell. So here we can see that this copy, or this homologue rather, originally was 100% maternal before crossing over happened. And in this alignment, we can see that the copy that is entirely maternal is going to go to the top daughter cell, whereas in the alternate alignment, if this chromosome aligned the other way, now the mostly maternal half is going to be pulled towards the bottom, and the result is two different daughter cells. Now, the reason why we use alignment rather than assortment is, remember that we're not pairing in a double file line. We're not deciding which homologue goes in which direction. We're deciding which copy is moved to which daughter cell, which is why we refer to this process here as what we call independent alignment. And independent alignment is going to decide which copy of each chromosome is going to be pulled to which daughter cell. And of course, the same possibility that exists, let me just write a clearer possibility. Remember that there are two possible alignments, one copy facing the top and one copy facing the bottom, or we can rotate the chromosome 180 degrees to get the other possibility. So this means that there are two possibilities per chromosome, and there are 23 individual possibilities, which means that the total number of possible different daughter cells that we can get is 2 times 2 times 2 times 2, equal to the number of chromosomes there are. So 2 times 2, 23 times, is the number of possible daughter cells that we would get just due to independent alignment, because of the fact that we no longer have quote-unquote genetically pure chromosomes, and the two copies of each chromosome that distribute to either end of the cell are going to be different. From this point, the process of cell division is basically identical to mitosis. So in anaphase 2, we can see that the microtubules are beginning to depolymerize, which once again means that they simply break themselves down in order to make them shorter, which pulls each copy of each of the 23 chromosomes towards opposite ends of the cell. So copies of each of the chromosomes move away from each other towards opposite sides of the cells. And the space here was quite short because we can use the word towards the poles of each of the, or toward where the poles where each daughter cell is going to form. And telophase is easily recognizable because we see the easily identifiable contraction of the metaphase plate that is happening. And of course, the contraction of the metaphase plate is meant to split the cytoplasm into two different daughter cells. This, of course, is the process of cytokinesis. And now that the different copies of each chromosome have segregated to opposite sides of the cell, we can see that the nuclear membrane is beginning to reform on each side. And once we have made it to this point, now the chromosomes can actually decondense, meaning that the DNA gets to unfold itself so that it is now accessible for the purpose of protein synthesis, because even though eggs and sperm ultimately exists to create a fertilization and a new offspring, that doesn't mean that the DNA is just going to be stored. The DNA of a sperm or egg cell is used to produce the proteins that are needed for an egg or sperm in order to survive, and now that chromosomal division, both to make sperm and egg haploid and to get the copy number down to one copy of each chromosome per cell is done. Now the DNA can actually exist to serve its function, but it can only do this if it opens up once again. Now, this concludes our summary of Meiosis 2, but it helps to take an inventory of everything that we have done so far. So as we can see, at the end of Meiosis 2, the two daughter cells that were produced at the end of Meiosis 1 have divided again, giving us a total of 4 unique daughter cells. And if we follow the color coding here, we can see that the homologs of each chromosome, as well as which copy was received by which daughter cell, are distinct. So these 4 daughter cells not only are more numerous than what would have been produced in mitosis, but are all genetically unique with each other. Now, because of independent assortment in Meiosis 1, they are also haploid, meaning that they only contain one maternal or paternal homolog each, and only one copy of each chromosome, because of course the other maternal and paternal homolog would be provided by the other gamete during fertilization. And at this point, we are ready for the sperm or egg to mature to undergo sexual reproduction. So to conclude this video, we can take an overall summary of what happens here. So after interphase, specifically after S phase and the duplication of DNA, our original parent cell is going to be diploid, meaning that our chromosome number is 23 times 2, maternal and paternal homologs of both, but because the DNA was duplicated in S phase, the copy number is 2, and so we have 2 copies of each. During the process of prophase 1, we get crossing over, where the maternal and paternal homologs undergo synapsis and swap pieces of their DNA to create hybrid chromosomes. In metaphase 1, we get the independent assortment of maternal and paternal homologs, which means at the end of Meiosis 1, we now have 2 daughter cells that are haploid. In fact, that is the key purpose of Meiosis 1. The problem is, as we see from the X shapes of chromosomes, that even though we have 23 times 1, we only have one maternal or paternal homolog of each chromosome. We have 2 copies of each of them, which necessitates meiosis 2, which more or less happens the same way that mitosis does, except during metaphase 2, because we usually have hybrid chromosomes. Depending on which direction the chromosome and its copy align on the metaphase plate, this is going to affect which copy goes up and which copy goes down, our final source of variation in the process of gamete production. And of course, by the time we reach the end of meiosis 2, we now have preserved our haploid daughter cells, but the copy number has been reduced to 1 because each individual gamete only has one maternal or paternal homolog and one copy of each, because the other copy were brought to the second daughter cell that is made per starting daughter cell that we started with in meiosis 2. To conclude this, you can test your knowledge of meiosis and the process of gamete production Firstly, by starting with this case study, which analyzes a number of genetic conditions such as Down syndrome that result from when meiosis does not actually occur regularly. Once you finish taking a look at this case study, you can try the practice problems below as well. And in the next video series, we're going to look at Mendelian genetics, which looks at using probability in order to predict how likely it is humans and other organisms will inherit specific genetic traits.